This is video number 3 in our series about myopia. In this, we explore how the eye controls its own growth. In previous videos, we have discussed what myopia is, both in terms of the well-known blurry distance vision and the less well-known risks it poses for eye health. In this video, we are going to discuss how the eye regulates its own growth, which works well for most people, resulting in good vision. However, one group escapes from growth control to become myopic. That divergence is the subject of video number 4, in which we explore what factors are associated with myopia. But first, we need to understand the basics of eye growth. This is the plan for this video. After a short review of myopia, we will discuss how the eye controls its own growth through the process of emmetropization. We will look at animal experiments in inducing myopia, how the mechanism was located to within the eye, and current theories on how it works. We begin with a brief review of what is myopia. If you watched video 1, you can skip ahead to the introduction of emmetropization. In the sense that your eye works like a camera, if you had ideal vision, light rays from a distant object would come to a focus and make a sharp image on the retina in the back of the eye. This ideal of sharp vision is called emmetropia. To the extent eye growth gets off track, the eye can become near or farsighted, usually hyperopia resulting from a too short eye and myopia from a too long eye. The amount the eye is out of focus is called the refractive error. The power and sign of the lens needed to bring the image into focus is called the refractive correction. Correction is measured in diopters, shown on the number line. Hyperopia correction is in plus numbers here to the left of 0. Myopia correction is in minus numbers to the right of 0. Regarding myopia, some studies specify that myopia formally starts at a spherical equivalent of minus 50 diopters. Others use minus 75. From there, it is divided into different levels, mild, moderate, and high, based on both reduced vision and complication rates. Getting control of the growth process is where emmetropization comes in. As the eye grows, the focusing power of the front of the eye gradually changes along a pre-programmed course. The mechanism for getting the image to land on the retina depends on fine-tuning the length of the eye. Here is what that looks like in emmetropes. The power of the cornea and lens gradually decrease, while axial length increases. In this case, neatly synchronized so the refraction remains stable near 0. To do that, the eyeball has a feedback system that gives it the ability to regulate its own growth based on the ability of the retina to distinguish an in or out of focus image, in this case, in the peripheral retina. Specifically, it speeds up or slows down length change to keep the incoming image in focus on the retina. What a fascinating piece of physiology. Let's talk about growth. Consider that your body and the organs that make it up are not random sizes and shapes. Rather, from person to person, they are all remarkably uniform. Take height, for example. Growth charts show how tightly regulated control systems are. Girls at age 20 have only a 14% spread of values from the 5th to 95th percentile. Regarding the human eyeball, here comparing relative sizes of newborn and adult. This is the growth curve for average eyeball length from post-conception up to 3 years of age. Birth is at 40 weeks. This is the length to height chart for boys for the same period, birth to age 3. Let's see how they compare. We extract eyeball growth from birth to age 3. Then we change the scale on the graph to percent growth based on birth dimension, showing the eye increases in length by about 1/3. That compares to growth in body length, which almost doubles in 3 years. Looking at the tightness of eye growth control, like we did with height, this is the range of refractive error at age 3 months. By 9 months, the gold line, refractive error has lessened and range has narrowed, a result of guidance by emmetropization. By adulthood, the spread of values remains narrow. In this study of 12,000 people, the average refraction was about plus a half diopter, with a standard deviation of 1.13. Another way to look at how well emmetropization works is to ask, what happens to people in whom emmetropization isn't working because they have some kind of pathology that degrades the retinal image, like cataract or retinal disease? The result, the orange line, shows that without a clear image, feedback control is lost, and the eye takes on a wide range of myopia, up to minus 14 diopters in this survey. Comparison to the narrow distribution of normals shows what an effective mechanism the eye has in aspiring to emmetropia. What a concept! The eye controls its own growth. By what mechanism could that work? How did researchers figure out where control is located, in the eye or in the brain? Does it respond to accommodation and/or light, or something else? A good part of our understanding of the mechanism comes from animal experiments. For several decades, researchers have been able to create myopia in animal models. From these experiments, we have learned the basic features of myopia development. Historically, the first animal model was found by accident. In experiments on vision development in monkeys, researchers sutured the eyelids shut of one eye to see what would happen to brain development. They noticed that the eye with obstructed vision grew much longer than the fellow unobstructed eye. This mechanism for development of a longer axially myopic eye is called form deprivation, or deprivation-induced myopia. A little later, researchers found they could also induce myopia or hyperopia with lenses. This is an example of a foam-mounted lens glued around the eye of a chick. They could create form deprivation by using a diffuser lens, which decreases image quality. Alternatively, they could manipulate refractive results by putting a refractive lens in front of the eye, creating an out-of-focus image, either myopic or hyperopic. For example, a minus lens creates hyperopic defocus, which causes the eye to lengthen, resulting in myopia. Both these techniques have been used in multiple animal models of experimental myopia. We note there is a difference between deprivation and defocus, which will come up later. First, we should note what animal refractive growth looks like, here shown in multiple animal species, including humans. Most animals start out hyperopic to varying degrees. Then, over the first 3 months of life, they progress toward emmetropia. One longer study of monkeys showed they maintain a stable refraction past age 4 years. The coming series of experiments is going to show how refraction can be modified as a way of figuring out how and why myopia develops. Here is a fairly recent experiment using diffuser lenses to develop form deprivation myopia in young monkeys. The experimenter outfitted each monkey with a pair of goggles. One eye had a clear lens, the other eye had the experimental diffuser lens. On the result graph, the x-axis is age, the y-axis is the difference in refraction between the treated eye and the untreated control eye. Discussion of the light effect comes later. In the left graph, the lines show the refractive difference between the two eyes over time. This graph shows the end result for each pair of eyes. Normal controls on the left. In the middle, you can see the preponderance of eyes with blurred vision became more myopic. So the poor quality image of form deprivation creates myopia. Why myopia? The answer is uncertain, but it is consistent across animal models. Besides form deprivation, the other mechanism of creating myopia is by using corrective/refractive lenses. This 2013 series is from the same research group. Again, each monkey was fit with a pair of goggles. One eye had a clear lens. This time the other eye had a -3 diopter lens, creating hyperopic defocus. The y-axis on each graph is the difference between the treated and control eyes. On the left graph, you can see how the refractive difference progressed, with relatively increasing myopia in the treated eye. The right graph shows the axial length difference increasing over the weeks of treatment time. Remember that increasing axial length is the physical mechanism of myopia creation. This is a clear demonstration that putting a minus lens in front of the eye induces myopia. An interesting question to ask is, what happens when you take off the experimental lens? If the eye does have feedback control, will this change the course of development? When the experimental lens was removed, the difference between the two eyes progressively decreased, meaning the treated eye indeed self-corrected toward emmetropia. The graphs show both the refraction difference and the axial length difference are decreasing. The experimental eye is recovering, telling us that emmetropization is still working even with the reset of input. Here is something unexpected, but important because it connects to other threads in this discussion. The amount of light available affects myopia development. How is that? In the experiment with form deprivation, the left graph shows a strong trend to develop myopia. The right graph shows a set of monkeys using the same lenses, but raised under bright light conditions. In this case, myopia development was significantly reduced, clearly seen in the end result graph. In this experiment using form deprivation, bright light mostly prevented myopia development. How about the group of monkeys with lens-induced myopia? The yellow lines are monkeys raised under bright light conditions. In this case, development of myopia was essentially unaffected by bright light, and the same with recovery after lens removal. Thus, in the monkeys with lens defocus, bright light did not affect myopia development or recovery. The Smith group, who did these studies, presents this summary graph. Controls are on the far left. In the form deprivation group, bright light significantly reduced myopia development. Whereas, in the lens defocus group, bright light had no significant effect on myopia development. Why this is so is currently unknown. It does leave us with the question of which model applies to the documented role of sunlight in myopia development. To show that emmetropization can go both directions, we look at one part of a set of experiments done in chicks, reported by Ashby. We will discuss the light effect later on. For this part, they took a group of chicks and put a corrective lens over one eye and no lens over the other. In one group, they put a -7 lens over one eye, rendering it hyperopic. The other eye was the control, the orange line here. The gold line shows how the eye with the minus lens became progressively more myopic, just as we saw in the Smith studies. They took another set of chicks and put a +7 lens over one eye, rendering it myopic. The orange line is the control. This time the gold line shows how the eye with the plus lens became hyperopic. This pair of experiments is a nice demonstration that emmetropization controls refractive development in either direction. The next question is, what is the mechanism of that control? The focusing power of the cornea and the lens decrease as the eye grows. They are little affected by the feedback process. That is, they seem to follow their genetically pre-programmed path. It is the change in the length of the eye that controls refractive development. We are focusing on development of myopia, with lengthening of the eyeball beyond normal. To start with, what part or parts of the visual system have feedback control of ocular growth? Does it come from the visual cortex, the midbrain, or the eyes? To understand the options outside the eyeball, for background we will briefly review the visual system and accommodation. Light is sensed by the retina. On the input side, the gold line represents a ganglion cell axon leaving the retina and traveling via the optic nerve to the lateral geniculate nucleus. From there, another axon travels to the visual cortex, where the brain interprets the arriving data in successive steps to create a conscious image. To sense an out-of-focus image and engage accommodation requires a number of areas in the brain. On the output side, the responsive accommodation involves 3 steps. This happens in both eyes together. For clarity, we will diagram just one side. 1. There is a signal to the ciliary muscle in the eye to contract. This makes the lens rounder to increase its focusing power, appropriate for reading and near work. It involves a synapse in the ciliary ganglion. 2. Another signal goes to both medial rectus muscles, causing the eyes to converge, so they are still pointed at the target object as it gets closer. 3. A signal goes to the iris to make the pupil constrict. Thus, the accommodative triad exists to help you see up close. Can we tell if any part of the visual system, input or output, controls emmetropization? We talked a while ago about the discovery that monkeys who had their eyelids sewn shut developed elongated eyes and myopia. That result inspired those investigators to dig deeper. In 1985, they published a set of experiments done in macaque monkeys investigating the mechanism of myopia development. This set of experiments is important, so we will spend a few minutes covering the highlights. For full details, you will need to look at their paper. In the first experiment, they fused one eyelid in such a way as to leave a translucent layer of lid skin, allowing light to enter the eye, but not form vision. The other eye was left open as a control. After 1 year, they did measurements on both the open and fused eyes. In terms of refraction, the eyes with the lid fusion became more myopic, as we have seen in other deprivation models. The open, normal eyes tended to be hyperopic. Then they measured eye dimensions to see what physical changes were associated with the myopia. The first 3, corneal curvature, depth of the anterior chamber, and thickness of the lens, represent the focusing power of the eye. In these dimensions, there was no difference between the open and fused eyes, so changes in the focusing power were not the source of myopia. The only difference was the depth of the vitreous chamber. In other words, the mechanism of myopia development was increasing length of the eye. The next question is, what location in the visual system is responsible for the length control? Is it the brain? Here we are specifically asking about the visual cortex. To test that question, they removed the visual cortex. Then, with one monkey, they fused the lids of one eye, and with another monkey, they fused both eyelids. The monkey that had monocular fusion closure, at 1 year, the open eye was +50, while the fused eye was -5. For the monkey that had binocular fusion, at 1 year, both eyes were significantly myopic, showing that even without a visual cortex, form deprivation myopia still occurred to the same degree. Next, they sectioned the optic nerve at the chiasm. The eye with the fused lids still turned out myopic. First conclusion: removing the visual cortex or sectioning the chiasm did not prevent deprivation myopia, meaning neither visual input nor visual processing had a role. Next question: is control related to the outgoing accommodation pathway? One intervention was to remove the ciliary ganglion, a key node in the accommodation pathway. Result: the fused eye was still myopic. Moving forward, does the ciliary muscle play a role? To test this, they applied atropine, a medication that paralyzes the ciliary muscle. They used 2 monkeys. In one monkey, they fused one side, instilling atropine in just that eye. Myopia still occurred. In the other monkey, both eyelids were fused, but they only put atropine in one eye. Both eyes developed the same amount of myopia, showing that paralyzing the ciliary muscles with atropine had no effect. This is an interesting finding in light of later treatment trials. In summary, in this set of experiments, they blocked both visual input at the optic nerve and visual processing at the cortex. Neither affected form deprivation myopia development. When they blocked accommodation at 2 locations, the ciliary ganglion and the ciliary muscles, that also did not affect form deprivation myopia development. There are a lot of details here. If you want to know more about these experiments, refer to the original paper. Other authors have experimented in the accommodation pathway. We will mention one. Working in chicks, Scheffel lesioned another part of the pathway, the Edinger-Westphal nucleus. Then they put on both plus and minus lenses, and the expected hyperopic or myopic development still occurred. We have spent a bit of time here because accommodation has received a lot of attention for its apparent association of near work with myopia. On the basis of these experiments, having blocked both the incoming and outgoing pathways of accommodation, that seems like strong evidence against accommodation as a mechanism of emmetropization. But not so fast. In video number 1, we presented a representative sample of large and well-done observational studies on factors associated with myopia. They showed agreement on the role of parents and outdoor time, but they showed conflicting results about association of near work with myopia. If accommodation is not the mechanism, what is? Our next set of experiments involves investigating features of emmetropization located within the eye. Question 1. Where is the controlling image located? The fovea is where we perceive image sharpness, and it is where we focus the image when we refract for glasses. However, the image that controls emmetropia is not from the fovea, but from the mid-periphery. An important point we will cover later on is that the peripheral focus may not be the same as at the fovea. We now look at a series of experiments that found the location of the controlling image. In 2007, the Smith group used a laser to destroy the central fovea in one eye. This is a cartoon of a normal monkey retina showing the region of the fovea. Here, using a laser to destroy the fovea. The results are presented in the same format as the previous Smith studies, comparing difference in refraction between the untreated eye versus the laser-treated eye. In the control subjects, the difference in refraction between the 2 eyes remained small through the test period, as expected. In the laser-treated group, the difference between the 2 eyes also remains small, just as in the control group, showing that absence of the fovea did not affect emmetropization control. To see what that looks like in terms of refraction, this is an individual monkey versus controls. Here the y-axis is refractive error. The thin lines are all the controls. For the individual test monkey, we have removed the control lines. The open circles represent the untreated eye. The dark circles represent the laser-treated eye. Back together, both the controls and this subject follow the same path in emmetropization, roughly starting out over 4 diopters of hyperopia and ending at around 2. Note the time course. Emmetropic correction happens within 3 months, then remains stable. Again, a missing fovea does not affect emmetropization. Then there is a complementary experiment obscuring the peripheral retina. Again, they fit monkeys with goggles. Both lenses were opaque with a hole or aperture in the middle, causing form deprivation for peripheral vision, limiting clear vision to the size of the fovea. Looking in concept something like this. You can see the complementary nature of these two experiments. Blue represents the clear image, gray indicates the degraded image. The results? The left and center graphs show the course of refractive error over the weeks of treatment. The graph on the right shows the end results, with the development of myopia, showing that projecting a blurred image outside the fovea causes the same result as other methods of form deprivation. Here is another view of the results following refraction change in a representative monkey. The thin lines are the controls, which we will remove for clarity. The blue bar is the period with the aperture lens in place. While the aperture lens is in place, myopia increases progressively. After the aperture lens is removed, allowing a clear peripheral image, there is recovery from myopia, heading back to normal development. That is additional evidence that it is the peripheral retina that is in control of emmetropization. The researchers took this experiment one step further. After the aperture lenses were removed, in this subgroup they used a laser to ablate the fovea. We again remove the control lines for clarity. With the aperture lens, myopia develops as we have seen. So what happens if you now ablate the fovea? Without central vision, can the eye still find its way back to emmetropia? The answer is yes. Without the fovea, the eye still recovers to emmetropia. Before we leave, there is another way of investigating locality within the eye. Instead of looking at the center and periphery, one can look side versus side. That is, put a lens that has a diffuser on the outer temporal side so that the image on the inner nasal side is degraded. Looking in concept something like this. Researchers can then measure the refraction and eyeball length at different angles off-axis. On the result graph, the x-axis is the eccentricity. To the left of 0 is the temporal side, which had a clear image. To the right is the nasal side, which had the obscured image. The y-axis is refraction difference between the two eyes. Here is the refraction result from a representative subject. The black circles are the eye that was treated, the white circles are the eye that was untreated. The nasal side, indicated by the gold bracket, is the side that is obscured by the lens. Over time, clearly it becomes more myopic, while the temporal side remains near the control. So, emmetropization control is further localized within the eye. This series of experiments confirms that emmetropization control is localized within the eye. Surprisingly, it is not in the fovea. Instead, it is in the mid-periphery. And the effect respects which half of the eye? Here is another big question. How does the retina know which direction the defocus is in? It responds to hyperopic versus myopic defocus and adjusts the direction of growth accordingly. There are a number of theories. I will describe one. So far, when we have discussed image formation, we treated the incoming light as if it all falls in sharp focus on the retina. But it's not that simple. For example, so-called white light is made up of multiple colors, namely equal amounts of red, green, and blue. If there's any doubt about that, go to my Color Vision series, or more simply, go to your paint program and look at the RGB makeup of white or any of the shades of the gray achromatic colors. We recognize that shorter wavelength blue light is bent more by a lens than longer wavelength red light. That means the different wavelengths come to a focus at different places. This is termed chromatic aberration. For our purposes, we show this occurring at the mid-peripheral retina. One theory about how the retina detects direction of defocus is by an ability to detect this chromatic aberration, more specifically red versus blue, focus/defocus. In this case, illustrating hyperopic defocus, the blue part falls on the retina so that part of the image is sharp, while the red part falls behind the retina so it is blurred. In this theory, the red defocus could be the signal motivating for a longer eyeball. This shows myopic defocus in which the red part of the image would be sharp and the blue part out of focus, motivating a decrease in eye growth. There are questions about this. For example, there are plenty of red cones, but are there enough blue cones to execute this? There are theories based on other possible clues, for example, image contrast, astigmatism, and other higher order aberrations. If you are interested, this is an extensive review. It turns out that light has an important effect on eye development. So let's talk more about it. Researchers have found that brightness, color, and duration of exposure all have significant influence on eye growth. We will look at several experiments on light effects. First off, the color of light can affect growth. To show the effect of monochromatic light, here is one recent study involving tree shrews raised in varying amounts of red light exposure. In a 24-hour day, there are 14 hours of light and 10 hours of dark. The number of hours of red exposure varied from none to all 14 hours. The exposure period was 13 days, and then they were followed after that. This is the control group, animals that had no exposure to red light, showing what normal development looks like. They started out moderately hyperopic and progressed toward emmetropia, ending slightly hyperopic. Adding exposure of 2 hours of red light per day, the red squares, slowed down progress toward emmetropia considerably. Interestingly, when the red light was discontinued, the eyes redirected their course toward emmetropia. The upper red circles show the animals that had 14 hours of red light exposure per day. During the red light period, their progress toward emmetropia was essentially arrested. Again, when the red light was removed, they redirected their course toward emmetropia, showing that color has a significant effect on growth. Next is the effect of the brightness of light. First, Cohen gives a straightforward demonstration of the effect of light without experimental lenses. In this experiment, researchers exposed chicks to 3 different levels of light, again with no experimental lens. High intensity is like outdoors on a sunny day in the shade. Medium intensity is like a bright room in your house, and low intensity. For medium intensity light, this would be the normal growth curve. The eye started out hyperopic and progressed toward emmetropia, as expected. Those in low intensity light progressed to myopia, like in form deprivation. What then would be expected with bright light? Exposure to bright light, the yellow line, slowed development, resulting in mildly hyperopic eyes. Thus, with no lenses, bright light slowed progress toward myopia. Now we return to the Ashby experiment, this time looking at the effect of light brightness on lens-induced refractive error. Here the orange lines represent the control eye without a lens, which stayed stable. Note here that the bright light did not affect refractive development. As we have already seen, the eyes with the minus lens, the gold dashed line, developed myopia. Bright light, the solid line, slowed the development of myopia. But the end result was the same. As we saw before, the plus lens caused development of hyperopia. Bright light sped up initial development of hyperopia, but in the end, the result was again the same. To recap, Ward showed us a strong effect of red light exposure slowing emmetropization. Cohen showed brightness of light changed refractive development in chicks without experimental lenses. Ashby, also in chicks, showed only a change in rate, but no ultimate effect on lens-induced refractive error, and no effect on the control. Smith, which we discussed earlier, testing in monkeys, showed light effect depended on the cause of induced myopia, form deprivation versus lens-induced. Other experiments show similar variability in the effect of light, with different colors and different exposure cycles. One also needs to be cautious in applying avian results to mammals. What could be the mechanism for the effect of light? We are not talking about the photoreceptor serving vision. We're looking for control of eye growth. Returning to the Ashby experiment, Part 1 tested light and lens-induced refractive errors. Part 2 is a different experiment looking at the role of dopamine. Diffuser lenses were used to induce form deprivation myopia. Then they compared the effect of light intensity with and without an intraocular dopamine blocker. The first two sets of chicks compared the effect of dim versus bright light on development of form deprivation myopia. In room light, the light blue bar, chicks developed about 12 diopters of myopia. Bright light, the gold bar, cut the degree of induced myopia about in half. When they injected a dopamine blocker into the eye, the orange bar, the effect of bright light was almost canceled out. With the control injection of ascorbate only, the darker blue bar, the effect of bright light was not affected. The interesting result here being the blockade of dopamine almost eliminated the effect of bright light. In other words, dopamine is a significant participant in the effect of light on slowing eye growth, at least for form deprivation myopia in chicks. A decade after that last experiment, Ashby returns with new collaborators to ask, how are form deprivation and lens-induced myopia different? And how is dopamine involved? In this three-step experiment, they first created both form deprivation and lens-induced myopia, then did injections of dopamine, and finally added blockers of dopamine receptors. Both form deprivation and refractive lens use produced about 4 diopters of myopia. Daily injection of dopamine, or a fellow agonist, reduced myopia development by about half. They are not using bright light in this experiment, just dopamine. Injecting the agonist and a dopamine 1-like receptor blocker did not affect the result. Injecting the agonist and a dopamine 2-like receptor blocker about negated the result, suggesting that both forms of induced myopia are moderated by dopamine 2-like receptors. What does that mean? Taking one step further into the dopamine realm, production of dopamine is known to increase in the retina with light exposure. It comes from dopaminergic amacrine cells. These are important interconnecting cells within the retina. Dopamine then interacts with multiple retinal cell types via both D1 and D2-like receptors. As such, it is involved in the eye's response to light. One current theory is that which receptor dominates is in control of the direction of growth. The direction, however, may vary between species. Thomson showed us in chicks, but other investigators using mice find the opposite result. If you want to read further about dopamine, this is a good recent review. What goes on within the layers of the retina that controls the physical changes of emmetropization? There is a cascade of signals that go from retina to choroid to sclera to control remodeling. The retina, by whatever mechanism, is able to detect which direction the image is out of focus, in this case showing hyperopic defocus. The retina generates a signal to the RPE choroid. The choroid then generates its own signal, which tells the sclera to remodel and in which direction to remodel. Some of the candidate signaling molecules are listed here, including dopamine, as we mentioned before, also retinoic acid, nitric oxide, and growth factors. In video number 2, we covered scleral remodeling and how, in myopia, it resulted in a thinner, weaker sclera, the cause of the longer eye and the complications. Before we leave this section, there is an alternative theory of emmetropization led by prominent vision researchers Jay and Maureen Neitz. The alternative scheme proposes the sharp-grow theory. Because we are running long, I'm going to present the several parts of this theory added at the end. As we come to the end of this video on emmetropization, what did we learn? Animal experiments have been a big help in trying to understand eye development, specifically the process of emmetropization. The leading current theory goes like this. We noted that most infants are born hyperopic and become less so as they grow. The focusing power from the front of the eye gradually changes along a pre-programmed course. The mechanism for getting the image to focus on the retina depends on fine-tuning the length of the eye. Emmetropization is the process that controls length to establish and maintain a sharp focus. Unexpectedly, it is the image from the mid-peripheral retina that controls emmetropization. In the growing eye with hyperopic defocus, the eye generates a go signal for increase in length. The same go signal can be triggered by using a minus lens to induce hyperopic defocus or by form deprivation. If the minus lens is removed, there is a recovery phase of slowed growth, returning toward emmetropia. For myopic defocus, the eye generates a stop signal and growth slows, allowing the image to move back to the retina. The same stop signal can be triggered by using a plus lens to induce myopic defocus. A red light or bright light may also generate a stop signal. In this model, go and stop signals are considered as separate pathways. However, it is also possible there could be just a stop signal with moderated activity. When the stop signal is more active, growth slows. When it is less active, growth increases. By whatever mechanism the retina detects direction of defocus, there is a cascade of signaling within the eye that directs the sclera to remodel, thus controlling the length of the eye and refractive status. To gauge the success of emmetropization, we return to graphs we presented earlier. Human infants who start out hyperopic grow toward emmetropia. In adults, the narrow distribution of refractive errors shows us that emmetropization is in general quite successful. However, when pathology interferes with the process of emmetropization, the result is a wide range of myopia. However, looking at the population as a whole can be misleading. Looking closer at refraction in childhood, at the end of early development, emmetropia dominates. but around age 6, something changes and myopia starts to increase. Following this further through childhood, most groups remain relatively stable while the myopes continue to diverge. Those graphs showing myopes as a group suggest all myopes diverge at the same time. But that is not the case. Myopia onset occurs at different ages. The earlier the starting age, the higher the resulting myopia level. And the divergence from emmetropia toward myopia actually begins several years before passing the myopia threshold. The big question is, what is happening? Why are the myopes diverging? In the search for causes of myopia, the leading candidates are family history, light exposure, near work, and peripheral defocus. Which leads us directly to video number 4, where we explore the divergence of myopes. We have finished the main part of our video, and we now enter an extra section. As we mentioned before, there is an alternative theory of emmetropization. This work is led by prominent vision researchers Jay and Maureen Neitz, who are best known for their body of work in color vision and genetics. For the standard theory of myopia development, we are calling it the blur-grow theory because the peripheral blurred image regulates growth. A hyperopic blurred image stimulates elongation. When the image becomes sharp, growth slows down or stops. An alternative scheme proposes the sharp grow theory. This is a summary as I understand it. The theory contains two elements. First, contrast detection by the retina is based on L and M cone function in the center surround sensory unit. In both theories, emmetropization is controlled by peripheral sharpness/contrast detection. Second, MYP1 is the first gene mutation identified that is known to result in myopia. It causes relative dysfunction of one cone population. The Neitzes asked the question, how does a color cone defect cause myopia? In the standard theory, a peripheral blurred image results in a balanced signal to the bipolar, which is thought to signal the eye to grow. The blur-grow theory. A sharp high contrast image sends an unbalanced signal to the bipolar, signaling it to stop. When one cone population is abnormal, even illumination causes unbalanced input, usually a sharp signal. They reason that since the known result is growth/myopia, that means a sharp image input stimulates growth, the sharp grow theory. This group goes on to propose a scenario of how the sharp grow mechanism could have evolved. A relatively young hyperopic person, when focusing up close, might under-accommodate, leaving the background in mild blur only. The sharper background signaling more growth. An older, less hyperopic person, when focusing up close, might fully accommodate, leaving the background in greater blur. The blurrier background signaling less growth. Connecting that to the recent myopia epidemic, in the modern world, young people spend a lot more time indoors, engaged at close and intermediate focus for reading, computer work, and video games. The Neitzes contend that in this environment, peripheral objects are at a similar focal distance to the center of focus, as in this illustration. Because the periphery remains in relatively sharp focus, that stimulates myopia development. This theory of emmetropization led them to develop a unique glasses design we will discuss in the treatment section. If you want to read more about this theory, the two top references are recent papers by major authors. If you prefer a video presentation, the last is a video of a grand rounds presentation by Maureen Neitz, one of the principal investigators. The Neitzes have a couple more interesting observations. It turns out the L to M cone ratio, red to green, varies in different populations. As a significant example, the ratio in Caucasians is about 1.7 to 1, whereas in Asians it is about 1 to 1. There is data correlating the L to M ratio with average axial lengths. When red cones dominate, there is more likely to be emmetropia, while if the ratio is even, there is more likely to be myopia. They cite this as one possible explanation of why myopia is more common in Asia than Europe or the US. Another observation involves the haplotype of the L and M opsins. These genes are on the X chromosome with only one active copy of each gene. Looking at exons 3 and 4, sequence variations in these exons determine how well the cone pigments function. The graph on the right shows that variations in these sequences have been found to be associated with specific levels of myopia. In 2015, based on these observations, the Neitzes filed a patent using both these approaches for diagnostic and treatment proposals. First proposal is to measure the L-M ratio and/or determine the haplotype to predict who would develop myopia. Second, use those findings to prescribe a lens with a wavelength-dependent optical filter, or prescribe blur-inducing lenses, either as contacts or glasses. The blur-inducing lenses are currently undergoing a 3-year trial, discussed in the treatment section. With that, we reach the end of video number 3 in our myopia series. In video 1, we discussed what myopia was, how it is increasing around the world, and what factors it might be associated with. Video 2 is a discussion of how myopia leads to vision-threatening complications like retinal detachment and myopic macular degeneration. Despite the eye's ability to control its own growth, around age 5 or 6, myopes diverge from other refractive groups which remain stable. There are a number of potential causes currently under investigation. Last is video 5 exploring the variety of treatment options available and how successful they are in trials and their use in practice.